Hello, everybody. Today I'm going to talk about our wheat heat project that we are working on at Agriculture Victoria in collaboration with colleagues at the University of Sydney. Heat is one of the major abiotic stresses limiting crop productivity, and heat risk is increasing and is expected to be more severe in the future because of global warming. In Australia, for example, you can see how temperature has increased across the country as comparison between the year 1919 and 2019. It was previously predicted that for every 1 Celsius degree increase, wheat grain yield will be reduced by 6%. The broad aim of the project is to develop a pre-breeding germplasm with more heat tolerance. To do this, our first task was to develop a new genomic prediction model that incorporates environmental covariates to improve prediction accuracy. The second task was to detect stable QTL for different agronomy, physiology, quality, and nutritional traits under heat stress. And the last task was to select best parents with their genomic estimated breeding values or GEBVs to breed superior advanced lines that can be incorporated into commercial breeding programs. The population size we used involved 2,967 lines, which consisted of Australian cultivars, previously reported materials with heat tolerance, including MR wheat derivatives, CEMET and ICARDA lines, Indian cultivars, and lines provided by ACPFG. The population also involved new lines that we developed every year from crosses between parents with high GEBVs, of which best lines were retained for the next year. Field trials were planted in Narrabri in New South Wales with 2 times of sowing, optimal in May, late in July to ensure higher temperature during antithesis to grain filling. A core set of 200 lines were distributed to 4 other locations across Australia, and all trials were irrigated to avoid interaction with water stress. We collected detailed weather information for the trials. You can see here the variation between weather variables between TOS 1 and TOS 2, as well as the variation of a grain yield between both times of sowing. We used heading and maturity days to define environmental covariates or ECs for each plot at 7 time points from planting to pre-heading, 6 days before and after heading, and another 7 time points post-heading to maturity. So why we used ECs? Simply because they give detailed description about the environments we are dealing with. For simplification, looking at this figure, using E only will tell you that it's from the Simpsons, but ECs will tell you specific information about it. Therefore, ECs can help us to select genotypes with high GEBVs and low interaction with environments, assuming that these interactions can be better explained by ECs. Twofold cross-validation strategies were followed. In CV1, we fully removed lines from all environments, and in CV2, we assumed sporadic missing across the whole data. Resampling for 20 times was done, and the accuracy was calculated as the correlation between predicted and actual phenotypes. What makes our approach novel compared to previously published genomic prediction models with ECs is that we depended on a subset of ECs that represent the variation among environments, which reduce the noise introduced by irrelevant ECs. The algorithm is simple. First, we ran principal component analysis for the ECs. Then we correlated ECs to eigenvectors and selected the ECs with the highest correlations. And finally, we built a covariance matrix using the selected or the prioritized ECs. We tested different thresholds for the correlation between ECs and eigenvectors. And we found that the highest prediction accuracy was achieved with heavily prioritized ECs at threshold 0.9. Compared to the standard G-blob model that takes into account genotype by environment interaction, we got around 0.19 higher prediction accuracy on average across all replicates. The next milestone of the project is to detect stable QTL for different traits under normal and heat-stressed environments. And because the data is unbalanced, we use meta approach, which can work in such cases. First, meta-analysis is a type of analysis that deals with summary statistics calculated on actual data set instead of actual data itself. For example, in genome-wide association analysis, it can be used to combine different single trait GWAS analysis applied on different traits or different environments. This is useful to increase the sample size and which increase the power of the GWAS analysis and can be applied when raw data cannot be shared between different institutes. In the example presented here, you can see that we have 3 different single trait GWAS analysis in which each one has a single specific significant QTL and a common QTL, common signal that didn't pass the significant threshold in any. After running the meta-analysis, you can see that the common signal became significant while the remaining one disappeared. Our population was genotyped with 90K and the SNPs were imputed to exome sequencing level to end with total number of 341,000 SNPs. And after combining all the trials, we ended with 13,959 phenotypic records for grain yield. On the right, you can find circular Manhattan plot, and each circle represents one trait and significant QTL were highlighted in orange. And a total of 107 QTL were detected for all traits, of which many have never been reported before. Another aim of our work is to increase the nutritional value of our lines. As we know, more than 2 billion people globally have micronutrient malnutrition, and the most common element deficiencies are iron and zinc. But the most economical way to improve the intake of these elements is to improve the biofortification of grains such as wheat by breeding cultivars with high element concentrations. The data we used involved 60 trials and we measured the concentration of 13 different elements. The genetic correlation results showed that these elements generally have negative to low correlation with grain yield, which complicate the selection for them because farmers usually care about yield to increase their income. Our results showed that these elements can be predicted with moderate to high accuracy, and we also detected several loci controlling these traits that are independent of grain yield. We also defined a set of 63 bromycin lines that have above average yield with high element concentration, which seem to be very important genetic resources for future crosses. 53 of these lines were derived from cultivar BBW 550 and were highly structured, as you can see in the figure. If we want to have a closer look on the top performing lines across years, you can see that there are 3 pre-breeding lines that outperformed all released cultivars and had much higher grain yield for the normal sowing time, and they showed high performance for late sowing time. On the other hand, high-yielding cultivars for the optimal sowing time showed poor performance on the late sowing time, such as EGA, Gregory, and Suntop. And the same happened for the best cultivars in the late sowing time, such as Anuelo, Lang, and Sanko. To sum up, genomic prediction successfully aided the development of high-yielding cultivars under normal and heat-stressed environments. Using environmental covariates after prioritizing them improved our production accuracy. We also detected several QTL across environments for different traits, and the concentration of micronutritional elements can be predicted with medium to high accuracy. Finally, I would like to acknowledge the following people for their contribution to the research I presented, and I hope to meet all of you in 2022. Thank you.